Welcome back to Chapter 9. This is Part 1B. In our previous part, we discussed the fact that thin filaments are composed of actin and thick filaments are composed of myosin when we take a look at the anatomy of skeletal muscles. What we're going to do in this part of the presentation is we're going to take a look at the steps that are required to form the neuromuscular junction and to initiate the sliding filament theory. All right, now I'd mentioned to you previously that something that makes skeletal muscle unique is that it requires nerve innervation for it to actually start the contraction process. And it turns out that that is just one part of it. On your screen, what you're looking at are the 4 steps that are required in order for skeletal muscle to contract. It shouldn't surprise you that nerve stimulation is the first item to be discussed. And after that, what you're going to notice is the response that's being generated, including step number 2, which says that you need to generate an action potential, which is an electrical current that will be starting off in the sarcolemma. The third part means that we're going to have to amplify that action potential and propagate it along the entire length of the muscle fiber and allow it to also penetrate to the inside of the cell through the use of the T-tubules. And our fourth step will be the release of intracellular calcium. Step 1 and 2 will occur at the neuromuscular junction, which will be the focus of the first part of this presentation. And step 3 and 4 is what we're going to be calling our excitation-contraction coupling. That is when we physically connect the electrical signal from our nerves to the actual contraction aspect, i.e. our cross bridges formation and our sliding filament theory between the thin and the thick filaments. All right, so just a really quick reminder, on the right-hand side, I have placed a very simple illustration of a neuron. We have previously talked about neurons when we did our histology section, and we discussed the fact that neurons will have dendrites on top that are able to receive the signal, and then they have a long axon that will basically transmit the signal away from the soma or the cell body. Now, what we see happening in the case of skeletal muscle is that the end part of the axon will branch out and hover very closely to a specific part of the muscle fiber. And that physical location where the axon and the muscle fiber meet, that is going to be called the neuromuscular junction, or NMJ. It also goes by the name of the motor end plate, but the most common one will be the neuromuscular junction. Part of what we have to do as we continue through our presentation is to take a look at the individual parts of the neuromuscular junction, and of course we're going to incorporate that as we take a look at initiating contraction within our skeletal muscle. All right. This is focus figure 9.1, and it's a really nice comprehensive picture. It basically shows you some of the steps that are required to get our muscles to contract. And if you look all the way on top of the picture, you're going to see that neuromuscular junction that I was mentioning to you on the previous slide. So you see the axon coming up from the top of the page, and you can see the yellow protrusions. Those are going to be the knobs or the ends of the axons, and as you can see, they're making almost very close contact to the actual muscle fiber that's been drawn out. And highlighted towards the middle of the image, you can actually see the synaptic knob, the large yellow knob, hovering over a little cleft that's been created over the muscle, and that right there is going to be our neuromuscular junction. So that is the structure that I was mentioning to you beforehand. And what we now need to do is we need to take a look at all the different events that are taking place that are going to excite our muscle and eventually cause it to contract. All right, so let's take away some of the numbering so that we don't get overwhelmed by everything, and let's work our way through it step by step. So the first thing to understand about this picture is that you're looking at the interaction between the nervous system and the skeletal muscle. And it's highlighted once again by that little box where you see the yellow knob, and that is going to be the axon terminal, the end part of the axon coming down from the neuron. And the bottom part, that is going to be our little synaptic cleft. That is the sarcolemma, the plasma membrane of the muscle fiber. And the entire structure, that is then going to be called our neuromuscular junction. Now, to start off the process of getting your muscle to be contracted, you might recall from one of the first few slides in this presentation is the fact that you require to have a nerve signal. So what we're seeing here for step number 1 is it says that the action potential will arrive at the axon terminal of the motor neuron. And in fact, what we can do is we can look up and take a look at the myelinated axon of the motor neuron, and we're going to see a few of the pink arrows pointing downwards, and that is basically representing the fact that we are receiving the nerve signal coming down the axon, and it will eventually get to the end of the axon, which is going to be by the axon terminal. So our first step is illustrated right here, which is that the nerve signal is coming down, aka our action potential, and it arrives at the axon terminal. Our second event that we're going to see happening is the minute that the action potential reaches the end of the synaptic knob, we're going to go ahead and have an influx of calcium. The calcium is going to be flooding inside of the axons, moving down its electrochemical gradient. Because the calcium is responding to an action potential, aka an electrical signal, we're going to be able to say that the receptors are voltage gated. Voltage gated means that they respond to a change in the electrical current. So when the action potential came down, they noticed that there was a change in the electrical current, and the receptors responded by opening it up, allowing the calcium to flow in. Our third step is going to release this neurotransmitter called acetylcholine. ACH to be abbreviated. So what you're seeing on your illustration right here, and you can take a look at the big red arrow that's on the right hand side, is once the calcium floods into the axon terminal, we see that there are little synaptic vesicles that were contained within the axon terminal that will migrate towards the bottom of the axon, and they will do exocytosis, meaning that they will release their content. And the content in this case is the neurotransmitter acetylcholine. Now, acetylcholine will leave the axon and will start to travel towards the junctional folds that you find within the sarcolemma, which are the curved bottoms, and it's looking for its particular receptor so it can bind with it. So here on this illustration, this is step number 4, we now see that the acetylcholine has found its receptors. Now, one thing to point out is the acetylcholine, when it finds its receptors, is now interacting with the sarcolemma of the muscle fiber. So the acetylcholine, the little green dots, started out in the synaptic knob of the neuron. They did exocytosis, which is that they exited out of the axon. They travel through the synaptic cleft, which is the little opening, and it found its acetylcholine receptors, which are being housed on the sarcolemma, or the plasma membrane of the muscle fibers. You're also going to notice that within the plasma membrane, you have what we call junctional folds. Those are the curvaceous of the sarcolemma, and the whole purpose of that is to increase the surface area so that you can place more receptors so that the acetylcholine can find the receptor quicker and you can move on to the next step of muscle contraction. I also want to point out that the neuromuscular junction, when you look at it, it is the meeting point between the axon and the sarcolemma, so the nervous system and the muscle, but they actually don't make physical contact. They hover ever so close to each other, and that little space that is generated between the knob and the sarcolemma, that is called the synaptic cleft. So they don't physically make contact, they hover, and that is the synaptic cleft, the little space in between them. And obviously the junctional folds are the infoldings that are going to increase the surface area that the sarcolemma have to place their acetylcholine receptors. All right. Now, once the acetylcholine receptors match together with the acetylcholine, the neurotransmitter, what we're going to see happening is that those particular channels, those particular receptors will respond by opening up. And when they open up, they're going to allow an influx of sodium and an outflux of potassium. So on the little insert picture that just popped up at step number 5, you're going to notice that your sodium goes in, your potassium comes out. And by manipulating the sodium and potassium flow, you're going to cause what we like to call an end plate potential. An end plate potential means that that particular part of the cell is going to get excited. It's going to increase its membrane voltage. And by doing so, it's going to generate a response from the sarcolemma in a very localized way. So that is then what we call it an end plate potential. All right, I'm just going to quickly do an overview of our 5 steps to make sure that we're all on the same page, okay? So our goal is to get our skeletal muscle to contract. And in order to do that, the first step is to go ahead and recruit a nerve signal or an action potential from the axon of a neuron cell. And when that action potential makes its way over to the synaptic knob, it's going to cause your voltage-gated calcium channels to open up, and calcium will flood into the synaptic knob. As calcium enters into the synaptic knob, it will stimulate little synaptic vesicles to release acetylcholine into the synaptic cleft. The acetylcholine, which is a neurotransmitter, will then travel over to the sarcolemma, where it will find its acetylcholine receptors. Once it binds to the receptors, these ion channels will open up and they will allow sodium to flow in, potassium to flow out, and because of this change in electrolytes, we see that the membrane has an increase in its overall charge. It is becoming more positive. It is generating a response. So we are going to go ahead and call that an end plate potential. So at this point, we have excited the sarcolemma, but in a very localized aspect, aka only where the receptors are, so we have generated an end plate potential. Now, before we continue on, because obviously we want to take this end plate excitement and spread it throughout the entire muscle fibers so the entire muscle can contract and it's not just like a localized response. Before we continue to the next step of that, I do want to point something out, which is that acetylcholine has an enzyme that is able to inhibit its action. It's called acetylcholine esterase, or it likes to be abbreviated with your ACHE. And what we see happen is if the acetylcholine esterase is introduced into the system, it will quickly break down any free-floating acetylcholine. It can also remove it from the acetylcholine receptor, and when it removes it, those ion channels will immediately close. So I just wanted to point this out. This is step number 6, just to kind of show you that acetylcholine can easily be terminated by the breakdown of acetylcholine esterase, which is an enzyme that usually is released when the muscle is done contracting and is getting ready to relax. All right, so at this point, what we see happening is that we have our sarcolemma, the plasma membrane of the muscle fiber, and the muscle fiber is polarized. It is excited because we have opened our ion channels and we have created an end plate potential. So now what we need to do is we need to take this localized excitement and we need to spread it across the entire sarcolemma. And in order to do that, we need to revisit our action potentials and we need to understand the importance of depolarization and repolarization. So remember, our whole purpose here is to generate an action potential, an electrical voltage, and excitability across the entire sarcolemma. All right, so take a look at this picture and go ahead and start off on the left-hand side, picture number 1. And what it's showing you right here is that we have an end plate potential, which means it's a localized excitation. It occurs at the neuromuscular junction when the acetylcholine matches with the receptors and the ion-gated channels opened up, allowing sodium to flood in, potassium out. Sodium at this point is flooding in at a much faster rate. And that is basically what corresponds with the excitation. So what's going to happen is we're going to get a wave of depolarization. And as the wave of depolarization occurs, what's going to end up happening is following that big pink arrow in picture number 1, the excitation, the action potential is going to start spreading away from the neuromuscular junction and throughout the entire sarcolemma. And as this wave of depolarization, this wave of excitement starts to spread, we're going to see that there are a group of other channels that are going to respond to this action potential. And because they're responding to an action potential, that means that they're voltage regulated. So as the action potential spread, what we see happening is that you can have depolarization occur, meaning that the cells notice that the cell membrane is excitable. It has a more positive charge than normal, and this will allow the sodium channels to open, allowing sodium to flood into the cell, causing the cell to become even more excitable, even more depolarized. As you can see in picture 2, at the same time that the sodium channels are open, the potassium channels are closed. So it's the influx of sodium that causes you to depolarize and increase your membrane voltage, increase your excitability. We will also discuss that eventually you will have to switch over to repolarization, and repolarization involves the closing of the sodium channels and the opening of the potassium channels. And when you open the potassium channels, potassium will come flooding out of the cell, and with it, it will decrease the membrane voltage. It will make the membrane more negative, and we can use that to restore the sarcolemma back to its initial state, back to its resting membrane potential. All right, so just to recap, take a look at your picture. Picture number 2, depolarization means sodium in, the membrane voltage goes up, so the membrane becomes excitable. Picture number 3 is repolarization. Potassium out of the cell, the membrane will go down, it will become more negative, and this is when we are trying to restore the membrane back to its resting membrane potential. Now, some of you might remember this graph from previous conversations. So this is our typical action potential. So let's just take a few seconds and let's familiarize ourselves. We see that on this graph, we are recording time versus membrane potential, meaning the electrical current that is flowing through the individual cell membrane. In our case, we're looking at the sarcolemma of our muscle fibers. And what we're seeing is if you want to excite the membrane, you're going to be doing depolarization, and that is due to the sodium channels being open, and sodium flowing into the cell. At a certain point, you will peak. That is when you have the height of your action potential. And what we see happening is that the sodium channels will close off and the potassium channels will open. The opening of these channels will allow potassium to flow out of the cell, do an efflux, and as the potassium flows out, the cell will become more negative. It will decrease its membrane potential. This is called repolarization, as the cell resets itself back to its original membrane voltage or its resting membrane potential. So in essence, what you want to do is, if you want to have your skeletal muscle contract, you want to send that nerve signal down. You want to go ahead and communicate it with the neuromuscular junction, and as the excitement spreads, you want to depolarize the entire sarcolemma so it gets all excited and gets more positive, and the cells will respond by starting to do their cross bridges, and the muscle will start contracting. And when you're done with that and you want the muscle to relax, then we need to repolarize, reset the cell back to its resting membrane potential, and we can do the repolarization by allowing potassium to come out of the cell. All right, so let's take a look at excitation-contraction coupling. And at this point, what we're trying to do is we're trying to connect the action potential that's flowing across the sarcolemma and get it to actually connect with the myofilaments that are inside the cell. So your myofilaments are your thin and your thick filament. So how do we take the action potential that's happening on the membrane surface of the muscle, the sarcolemma, how do we communicate that to get the thin and the thick filaments to do their cross bridges and their sliding filament theory? Well, this is where your T-tubules come in. As you might recall from our previous lecture, the T-tubules are the infoldings or the invaginations. They are the little tubes that will connect the outside to the internal workings of the cell. So what we see happening is the action potential will spread across the sarcolemma, and when it comes across a T-tubule, it will allow the action potential to enter into the cell. And as it enters into the cell, we see that the sarcoplastic reticulum, the SR regions that sit on either side of the T-tubules, remember your triad? They will start to release calcium in response to all this excitation that's going around. So calcium starts to flood the inside of your muscle cells. So here it is an illustrated example. We are trying to show our excitation-contraction coupling. So you can see, if you take a look on the left-hand side, you can see the little purple arrow spreading the action potential from its original start by the neuromuscular junction, the yellow structure, all the way down the sarcolemma, and it's trying to find its T-tubule so that it can spread the excitation from the outside of the cell to the inside of the cell. Let's take a look at this picture a little bit more. It's more enlarged from what we originally saw. So starting on top, we can see that the action potential indicated by the purple arrows is spreading across the sarcolemma, and when it comes across a T-tubule, it will enter through the invaginations, which means it enters into the cell, and the minute it enters into the cell, the action potential is also noticed by the SR region, the sarcoplastic reticulum, which are the two blue regions on either side of the T-tubules, and as a result, you see lots and lots of calcium being released inside of the muscle cell. Now also notice that on the inside of the muscle cell, that is where we have our thick and our thin filament, and that's where the contraction part of the excitation-contraction coupling comes in, because now we have connected the excitation from the outside with the contraction that's about to occur on the inside of the cell. And in order for this contraction to occur, remember, our first step, as I said many times, is to make that cross bridge, that physical connection. Now, some of you might also recall the fact that the actin active sites are usually covered up by tropomyosin. Remember that long string protein that was covering the active site? So what we see happening is that we need to move the tropomyosin out of the way. And who's going to move it? Well, troponin, because troponin sits like a little knob on top of the tropomyosin and is able to move it away or over the active site. But the only way tropin will cause the tropomyosin to move is if it's exposed to calcium. So what we see happening is, within a few seconds of calcium being released from the sarcoplastic reticulum, we see that the calcium will make its way over to the troponin. It will be detected by the troponin, and the troponin will respond by moving the tropomyosin away from the active sites on the actin. So go ahead and look at your picture on the right-hand side. Look at the little insert, and for step number 3, it says calcium will bind to the troponin, and it removes the blocking action of the tropomyosin. And when it does so, it will expose the binding site so that the head of the myosin can interact with the actin on the thin filament. And then we have our cross bridges, our physical connection that has occurred. So I cannot overestimate the importance of the whole calcium release. So here we're just going to take a really quick break. Let's review some of our notes. Let's make sure we're all on the same page. So this is a quick overview of initiating our muscle contraction. So thus far we saw that the nerve impulse traveled down the axon of the motor neuron. When it reaches the axon terminal, we see that the voltage-gated calcium channels will open up and calcium will flood into the axon terminal. This will then cause the synaptic vessels to release acetylcholine into the synaptic cleft. Acetylcholine will travel over to the receptors and it will cause an end plate potential, i.e. an excitation to occur because of the opening of the sodium and the potassium, with a heavy emphasis on sodium entering into the cell. And as this end plate potential starts to spread, we then see that the sarcolemma will activate voltage-gated sodium channels, and these channels will open up, sodium will go flooding in, you'll have depolarization that will occur on a much larger scale, and you start initiating an action potential. So that's what this picture right here was trying to show you. And I know it might seem a little bit redundant, but you definitely want to keep an eye on every single little step that's occurring and get familiar with it. So take your time as you're going through your PowerPoints and as you're looking at the steps. We also talked about the fact that if you want to now do your excitation-contraction coupling, you want to get your thin and your thick filaments to interact with each other and make a cross bridge. We also know that we need to rely on calcium to do that. So if we're looking at our slide right here, it says if you have low levels of calcium on the inside of the muscle cell, that would then mean that tropomyosin will continue to block the active sites on actin, your thin filaments, i.e. the myosin head of the thick filament cannot make a cross bridge. And if we cannot make a cross bridge, then the muscle fiber is relaxed. As we see that the voltage starts to change because action potentials are being generated on the sarcolemma, and these action potentials protrude through the T-tubules, they will start to release calcium from the sarcoplastic region, SR region. The calcium will then cause the intracellular concentration of itself to increase, obviously, because it's being released inside of the cell. The more calcium is released by the SR region, the more likely that calcium will bind to troponin. Troponin will cause the tropomyosin to move away from the binding sites, and this will then allow, excuse me, the myosin head to make a physical connection to the actin. So the thick filament will make a physical connection to the actin, and now you have your cross bridges. Now, the cross bridges will continue as long as you have your calcium. The minute the calcium is taken away, then the troponin will go ahead and cause the tropomyosin to slide right back over the active site, thereby inhibiting the physical contact of the cross bridges to occur. So here we have our picture again. Once again, I know it's a little bit redundant, but we have to work our way through it. So we're doing our excitation-contraction coupling. We're going to take our action potential from the outside of the sarcolemma, fold it into the T-tubules, cause the release of the calcium. Calcium will go over to the troponin. Troponin will move the tropomyosin out of the way, and now the myosin can go ahead and physically bind to the actin, and you have your cross bridge. Now, when you have your cross bridge, remember, we're just initiating the contraction. So far, we've just made contact. We still need to slide over each other because the actual sliding mechanism is what's going to cause the muscle to contract. So, we have our cross bridges that are formed, but now we need to do another step, and this is going to be called a power stroke. A power stroke is when you lift the head of the myosin and you basically pull it towards a secondary binding site on the thin filament. And as it finds the secondary binding site, it pulls the thin and the thick filament over each other. Think of it as that you are on a boat and you have to pull up the anchor by yourself, right? So the anchor is attached to a rope. So as you're standing on the edge of the boat, you're going to go ahead and you're going to reach for that rope with one hand. And as you pull it with one hand, you're going to take your second hand and put it down the length of the rope, and you just keep going. So you pull the rope closer to each other, every time lifting one hand to move it to a secondary location, and pull the rope closer and closer. That's the same thing that's going to be happening when you do a power stroke. The myosin head is going to remove itself from its original position on the thin filament. It will cock its head and it will find a secondary binding spot to the thin filament, so it can slide the thin and the thick filament past each other. So on your slide right here, it says that we have 4 steps involved. We have the formation of our physical connection, our cross bridge, which is the myosin head attaches to the actin thin filament. We will then see that we have our power stroke, which is that the myosin head will pivot and go and bind to a secondary active site, allowing the thin filament to be pulled. We will have a detachment of the cross bridge, which will allow the myosin head once again to break that physical contact and move over to a secondary action site. And the cocking of the head will allow it to reach over to that secondary site that it's aiming for. Notice as you're perusing through the slide, you keep noticing that it keeps saying ATP. Chemical energy is going to be a huge factor to maintain this contraction aspect of the skeletal muscle. You will always require ATP throughout your cross bridge and power stroke formations. All right, I think this picture says it a lot better than I will in my explanation. So go ahead and take a few seconds and take a look at this illustration. And starting on top with the picture labeled number 1, it shows you the myosin cross bridge. So it shows you the connection point between the thick and the thin filament. And then the second part on your right-hand side shows you the power stroke, where the ADP and the phosphate are released and the myosin head starts to bend. And as it's bending it, it's going to start pulling the thin filament away, and it will then remove the head from the actual physical connection. So that's step number 3. It will require its ATP molecule, and finally in step number 4, it will cock the myosin head, and it will go ahead and attach itself to a secondary site on that thin filament, all while increasing the overlap and sliding the thin and thick filaments. And as it's doing this, what we see happening is that the Z discs are getting closer and closer together. The sarcomere is contracting, aka the muscle is showing its contraction as well. Here's another illustration, just basically showing you the same step, but obviously from a different way. This is from a McGraw-Hill book that we're looking at, and we once again see that the action potential, ATP is required to not only make your cross bridge, but also for the power stroke to occur. Now in this illustration, the reason I liked it is because you're going to notice that on the thin filament, which is represented by the little red beads, they went ahead and they colored one bead pink and the other one orange. So you can clearly see how when the myosin head is power stroking, it first makes a connection with the pink bead. It will then remove it, it will do its power stroke, it will cock its head and make a connection with the orange bead, and then it will drag it over. And as it's dragging it over, you're increasing your contraction because you're sliding your thin and your thick filament together. And here's just a closer look at a power stroke, which literally means that you alter the location of the connection between the thick and the thin filaments, and you allow the myosin head to glide over the thin filament. Now, a few things that I want you to think about as you're kind of perusing through this particular part of the notes. In order for your cross bridges and your power strokes to continue to occur, you will always need to initiate it based on the action potential from the nerve signal. You will always need calcium because calcium is going to allow the troponin to move the tropomyosin out of the way so you can make your cross bridge, and you will always require ATP. You need that chemical energy to maintain this particular part of the muscle contraction. If you would like your muscle to relax, you can do that several different ways. But usually the muscle will naturally relax when the action potential stops coming down the axon, meaning that the nerve signal stops. And when the nerve signal stops, then the entire steps that we looked at basically cease, Acetylcholine is no longer being released. In fact, what's remaining is being broken down by the acetylcholine esterase. The voltage-gated channels will no longer be responding to the excitation. They will start initiating repolarization. And as repolarization occurs, it will no longer amplify the action potential down the T-tubules, which means that the sarcoplastic reticulum will stop releasing calcium, and it will actually start increasing an uptake of calcium. So it will start removing the calcium and bring it back to storage. And as the calcium is removed, so does the troponin no longer respond, meaning it moves the tropomyosin over the binding sites of the thin filaments, and the power stroking and the cross ridges will come to an end. If you guys have ever heard of rigor mortis, it's a good example of what happens when the body runs out of ATP. So rigor mortis is stiffening of the body. We usually see this when a patient becomes deceased. Within 3 to 4 hours of death, we usually notice that they'll have a stiffening action where the fingers and the toes will kind of curl up, almost like the fingers are contracting and they can't relax. And that's exactly what's happening. It turns out that when the body dies, we stop producing ATP. And because we don't have any more ATP, we can't maintain that contraction and relaxation. So we see that the muscles will basically just kind of freeze in place. The rigor mortis usually peaks at around the 12 to 24-hour mark. It really has a lot of factors that go into it. It won't stay forever because eventually the myofilaments, meaning the thin and the thick filaments, themselves will start to fall apart. And when this happens, the muscles will naturally relax, and the coiled up action of the toes and the fingers will also go away, and the body will no longer be in rigor mortis. Please take your time and review these notes. And like I said before, we have our discussion sessions that are available, and I'm always an email away. All right, I will see you in a little bit for part 2 of chapter 9.